The next speaker is Dr. Janani Janani did her PhD in Institute of Mathematical Sciences, Chennai, in the computational biology group. I was fortunate to have Janani as my first student, and she was unfortunate to have me as her advisor. So she actually established in my group the work on linking environmental chemicals with adverse outcomes, especially health outcomes in humankind. Janani received recently the Outstanding Thesis Award from the Homi Bhabha National Institute across the different constituent bodies in the life sciences. So she has moved on to a position as a research scientist in pharma giant Novo Nordisk. So thanks, Janani, for finding time to come here and present your work.
Thank you, sir, for the nice introduction. Hi, good morning, everyone. I'm Janani, so I'm working as a research data steward in Novo Nordisk now. But here I'll be presenting about my PhD thesis, which I did here at IMSC. And my PhD thesis was more about exploring the harmful chemicals potentially to humankind. So So human health, it is determined by different factors. One will be genetic factors, and other it could be environmental factors. So the factors could be micro, medicine, pollution, or even the cosmetics that we use in our day-to-day life. So all these environmental factors are together called as exposehome, and this term was specifically introduced because the genetic factors are just not important for determining the human health. We also have to consider the environmental factor around us, and this can vary among different individuals depending on where they work and live. So what is the need for exposome research? So here I hope you can see the letters. So this is the chart. Like here you can see a list of diseases, and it shows how much of genetic variation is involved in each of these diseases. And here we can see at least 50% of diseases are not just determined by genetic factors, it also majorly influenced by environmental factors. So it's very important for us to study both, like including genetic and environmental factors. And also in this chart you can see that different levels of exposure risk factors that lead to chronic mortality, and still 49% of exposure risk factors are yet to be explored. Here during my PhD I was investigating the environmental chemical exposures, which is also called chemical exposome. And the environmental chemicals are important for a contributor for major global disease burden, especially on currently nowadays. And there are more than 100,000 chemicals in the market, and mostly they were not undergone toxicity testing. So now recently there is an immense research interest around to use network science and computational approaches to see how these chemicals can impact our human health. So that's how I studied in the PhD. So I wanted to see how these chemical exposome have been linked to the human health using computational approaches. And these are the outline for my talk. So first I'll tell you how we identified the environmental chemicals of concern to human health, and second how we have looked at the biological responses upon exposure to these chemicals, and third I'll be talking about the structural properties of these chemicals, and third we'll see how well these chemicals are regulated. And so in my PhD I have been studying all these different types of environmental chemicals, which includes endocrine disrupting chemicals, neurotoxicants, human milk contaminants, the chemicals which are already present in human tissues, and also the fragrance chemicals which are present in children products. So for this talk I'll be just focusing on the endocrine disrupting chemicals. And this endocrine disrupting chemicals, already the name suggests that it is affecting the endocrine system by altering the levels of the hormones in our body, which is very essential for normal functioning of a human body. So to identify these endocrine disrupting chemicals, which I'll be calling it as EDCs, so we have built a 4-staged workflow just to capture the EDCs with experimental evidence for endocrine disruption. So here first we started with curating the literature from different resources. And then we just want to focus on the experiments. So we have just considered the in vivo and in vitro experiments which was performed on humans or rodents. And then finally after following this 4-stage workflow, we were able to identify 686 EDCs in the first version of the work which we built. And for this I have curated around like 19,000 articles, and finally the 686 EDCs were identified from 1,796 articles. And also along with these EDCs I have also captured the endocrine mediated endpoints, that is what are the adverse effects these chemicals can cause, along with the dosage information, like at what dosage these chemicals can lead to such adverse effects. And once the endocrine mediated adverse effects were found, then these were again classified into different systems level perturbations based on what system it gets affected, for example neurological, metabolic, hepatic and immunological, etc. So and upon analysis we find that EDCs are more likely to affect the reproductive system than any other systems. And further these EDCs were further classified into based on their environmental source, chemical structure and also the literature evidence, that is it could be in vitro human, in vivo human or in vivo rodents. And finally we have also built a database which contains all these information, which is called DIDECT. And also just to highlight, this workflow was recommended by the French governmental agency for to capture such endocrine disrupting chemicals. And secondly, I'll just move on to like how we have looked upon the biological responses that are triggered by these chemicals. So when we are exposed to a chemical, so it can lead to series of effects. So first it can start from the molecular effect, then it can go to the there will be the cascading effect here, then cellular, organ, individual and population level responses. So we wanted to capture the responses at all these levels. So that's how we have leveraged a concept called AOPs, which is adverse outcome pathways. So here in this figure you can see this is the adverse outcome pathway, and this pathway will have key events and key event relationships, that is these are the key events, and the ones which are connecting are key event relationships. And the key event which is observed at molecular level is called molecular initiating event, and the one which are observed at organism level or adverse outcome. So this AOP framework can be used to organize the existing toxicological information from the literature to understand further the biological functions, and also to see at what level of biological organizations these chemicals can trigger responses. This AOP, so the AOP which I showed is like a linear network. So, but when these AOPs have, when there are different AOPs and when they have a common key event, this can be combined into a network. So it can form AOP network. So it can form into a complete AOP network. Then further we can study this AOP network based on specific research questions. So here I am just studying for, so these networks are called derived AOP network. So here I have studied for endocrine disruption. So we tried to construct a derived AOP network for endocrine disruption. So these are the steps on how to construct the derived AOP for endocrine disruption. So first we have extracted data from AOP wiki, which is a public repository which contains list of AOPs, key event relationships, etc. And this also contains some stressors information, which is the chemical stressor or non-chemical stressor that can lead to all these events. Then secondly, we wanted to, since the AOP wiki is regularly being updated, we just want to capture the information which are complete. So we have a set of instructions, and then we try to follow it to get only the curated data. So finally, that resulted in 161 AOPs along with 635 key events and key event relationships. Then we wanted to look into something specific to endocrine disruption. So for which we have manually curated the key events which we have captured, and we checked for some of the keywords like endocrine glands, endocrine hormones, receptors, disorders, or even the endpoints which we have compiled in DEDUCT. Then finally this resulted, So this resulted in 48 AOPs finally. These are these I would be calling as ED AOPs, that is the endocrine disrupting AOPs. And finally, we try with those 48 AOPs based on the common key events, we tried to build a ED AOP network, that is endocrine disruption AOP network. So, and here we can see that there are two largest connected components, and these are these two largest connected components are with 12 AOPs. So we tried to dig deeper into these largest connected components to see underlying mechanism of endocrine disruption. So here I will be illustrating with one of the largest connected components. So this is the expanded version of one of the largest connected component, and here you can see the list of key events, and which also includes the molecular events which are in yellow color, and the green ones are the key events, and the pink ones are the adverse outcomes. And the highlighted in the red are the common key events between two or more AOPs. So once we have seen this, we have also tried to overlay with the systems level information. So we have mapped to the different systems. For example, impaired fertility is reproductive system. So and also increased cell proliferation in hepatocytes can be hepatic system, and also we want to see how the endocrine system is affected, like once it is disrupted, how this is two different system levels at systems levels, how they are getting disrupted. So for this we have studied the point of convergence in this network, and also the point of divergence. So the point of convergence are the ones which where two or more key events go and converge into one specific key event, and this can be of for example here this is increased cell proliferation in hepatocytes is converged by 3 key events, and this can be further used to test some for risk assessment of EDCs, and these are the ones highlighted in yellow are the point of divergence, and this shows where the key events diverge from one system to another, and it help us to understand the mechanism even better. So this understanding this network topology is, helps us to find the biological association within the endocrine system, and it helps us to understand the endocrine disruption mechanism better. So then the third one is I'll talk about how we studied the structure of the environmental chemical space. Here, so we have built a network called chemical similarity network of EDCs, and here in this network you can see the nodes are EDCs, and they'll be connected by an edge if they share a similar chemical structural property. So to compute the structural similarity, we have used an index called Tanimoto coefficient, and this score generally ranges from 0 to 1, and 0 means no similarity and 1 means high similarity. And so after we have constructed this network, we try to see how well are these distributed, and we can see that there are 225 EDCs in this largest connected components, and there are many EDCs which are isolated, which means they don't have any much of structural similarity. And we find this, the chemical space of EDC is diverse, but however some of the modules have shared like similar structural properties. And then we want to see if the chemicals share similar gene targets. So here the nodes are the EDCs and they are connected by an edge if they share a similar target gene. So for this also we have used an index called Jacquard index, which is used to quantify the similarity between the target genes here, and we have observed the similar pattern like in the CSN, that is the chemical similarity network. So this clearly says that even though the EDCs can have similar structure, they may not share a same target gene. And then we wanted to find the correlation between the structural similarity and target similarity, because most of the predictive models are based on QSAR, which is quantitative structure relationship. It mostly relies on the fact that if the chemicals have same structure, then they will have same function. So we wanted to verify this with this data. And we find that the chemical similarity and the target similarity correlation is really poor, which really underscored the challenges in developing a model for EDCs based on their structures. So lastly, I'll be talking on how these chemicals are regulated in the market. So recently we have seen that there are so many literature which are growing on EDCs, but still we wanted to see how well it is impacted in the regulatory aspects. So for this we have compiled EDCs from different databases, including the DEDUCT, and then we have also compiled 36 chemical lists which are part of regulations and guidelines. So, and these 36 lists were further divided into substances in use and substances of concern. So, the substances in use are the ones which are already in use, and also the chemicals are were said that it is safe to be used. And also substances of concern are the ones which are already marked by the regulatory agencies as hazardous ones. And these chemical lists were further classified into based on their categories, like the use categories like biocides, cosmetics, environmental and water quality, food additives, etc. Then we tried to find overlap of these EDCs with the substances of use chemicals and substances of concern chemicals, and we find that there is a huge number of overlap, and these were further classified into 3 different groups. That is, the group 1 will only have the EDCs which are present only in substances in use, and this is group 2, that is the EDCs which are present in both SIU and SOC, and the EDCs which are present only in SOC or group 3. Then we tried to see how the numbers are distributed across different use categories, and also we tried to see how many chemicals are currently produced in high volume. And we see that the EDCs are almost present in all the use categories, and also they were also produced in high volume. This clearly shows the bridge between the academic research and also the regulatory aspect of these chemicals. These are some of the papers on my EDC, and also these are the other papers which are on the other chemicals, which also gave very interesting results which you can refer to. So since these chemicals are of societal concern, I think it attracted some of the media attention, both national level and international level. And finally, I thank my thesis advisor and also my colleagues for the support, and also most importantly IMSC for the support. So questions?
Oh, thank you. Thank you, Janani, for this nice talk. Quick time for 2 questions. So
Hi Janani, very nice talk. I was just curious, how do you define the Tanimoto coefficient to basically measure the similarity of chemicals? I'm curious.
So we use a fingerprint. So the chemicals has fingerprints called ECFP4 and also the max fingerprints. So it's like a binary fingerprints. So if it has some benzene ring, then it can be 1. So if it has some other attachment like functional group, then it can be 0. So it checks for such fingerprints, and there will be a binary fingerprints called ECFP4. So we have also checked for other fingerprints also. So it just matches with this. It's just intersection by union of those fingerprints.
So it's not like a Hamming distance, right? It's basically
yeah, it's like that.
normalized.
Nice talk. So the question here is the correlation that you did between the structural similarity and the target similarity, because like what you said, the SAR studies that medchem people focus on is based on the structure and what is the most important motif and how they can retain it and then create new analogs, right, to improve the efficacy. So if you're saying that that is untrue, what do you think is that underlying thing that so many chemicals have worked in the market, and is it because that there is a limitation currently that your target similarity is only looking at gene level and not protein-protein interactions, where so many of these chemicals might be playing a role at the protein level? What do you think about the low correlation?
So I think that we don't find a pattern because the EDCs, they are very structurally diverse first of all. So maybe the gene, as you said, yeah, it may vary when we are looking at the protein, but still we find that the structural diversity is more within the EDC group. So maybe we should consider some other factors also into account to build a model for EDCs. Like maybe we have to consider some of the adverse outcomes which we already have or something like that. So some different methodology should be there for the EDCs, I guess, even if we are looking at the protein level.